Okay, so this talk uh, is called A Rabbit Hole of Determinantal Identities, and you'll see the connection to Alice in a little bit. Um, I it's going to be maybe 2/3 expository and 1/3 research update. And to motivate this, you have to know a little bit about Emily Simmons' project in collaboration with Josh Pusey. She's presented on this in Biomath recently. So I'm not going to go into a whole lot of detail at this point. But basically, part of what Emily is going to be trying to do, her project has an empirical and a theoretical component. And in the theoretical part, she's trying to understand the way in which novel phenotypes can arise through hybridization. The system is Mimulus, monkey flower, which is commonly used in evolutionary and ecology studies. And what you're seeing in the top frame is a picture of two varieties of Mimulus that don't have much of a pattern, at least on the top petals. But when they are hybridized, you see a very different sort of pattern emerge. When you further across and look at what's going on in the F2 generation, you see lots of variation, and that's illustrated with some of the examples in the bottom square. Now, as Emily has tried to think through how this might be modeled, we've definitely started at least with a Turing type pattern formation mechanism, and the logic of this is needs to be explained a little bit. So what the idea is that we want to, in a normal Turing type system, you have an activator and an inhibitor, right? And so here that's species, chemical species U and chemical species V. But we have four partial differential equations, because the organism is at least diploid. Okay, so for argument's sake, I'm going to just, even though these are polyploids, let's just think about a diploid organism. And that would mean that, the gene product that was the activator, which is the letter U here, and the gene product that's the inhibitor, the letter V, there's two copies of them, one coming from presumably two different autosomal chromosomes. Now the cupreous variety and the variegatus variety, for different reasons, can be thought of as being homozygous, right? So they're going to have the same activator and the same inhibitor, but in their situation, they're not generating a rigorous pattern. However, in the F1 cross, there is a pattern that emerges. So in the F1 cross, the flower is heterozygous, and now you really do need the four equations. So for the homozygous case, you could have contracted this system to get a smaller 2x2 system, just using the fact that you had the two equations for the activator and the two equations for the inhibitor were identical. But I think it's easier to sort of just think about things from the perspective of all of the organisms are diploid. The issue is that in the case where you're homozygous and you're trying to think about the stability, the instability that's leading to the pattern, you have certain symmetries in the model parameters, right? So when you're homozygous, you have certain symmetries in the model parameters, and that's illustrated here with these arcs that are connecting various elements of the Jacobian matrix. So the Jacobian matrix is going to determine whether The system, the partial differential equations are unstable with respect to spatial perturbations in the presence of diffusion. It's a relatively simple mechanistic model, just an activator or inhibitor, but there's two of them because they're diploid. When you're homozygous, you've got these symmetries that are occurring. When you're heterozygous, you don't have the symmetries. And so that basically...
Greg, can I ask several questions?
Sure can. So let me just finish. Well, go ahead.
Okay.
Ask the question now.
Okay, so I remember when Cece was modeling this a couple of years ago, do we actually know what these activators and inhibitors are, or what gene they are?
Yes, there's a fair amount that's known about this. And so in my talk today, I'm going to be pretty generic, but we have a different level of modeling that's going on that's actually keeping track of things like that the transcription factors are basic helix, loop helix proteins, and one of them, the activator, is actually functioning as a trimer. There's two additional proteins that it interacts with before it's an active transcription factor. There's an inhibitor that's called, I think the activator is called Nigan. The inhibitor is called red tongue. So there's a fair amount that's known.
And given that these are inside cells, are the activator and inhibitor themselves diffusing outside the cell membrane and going through the petal, or what are they doing exactly?
Right. So for the reaction diffusion framework to make sense, we need these gene products to actually be diffusing from cell to cell. And that actually occurs in plant cells because there's a specialized junction in plants. They're called, Emily? Desmosomes, is that right?
Plasmodesmata.
Plasmodesmata. Okay, so they're sort of like gap junctions, but they're even bigger and they're specialized. It's something that only occurs in plants.
And if you look at oh, sorry, go ahead.
It just connects the cytosol between the plants so that proteins can flow pretty easily between plant cells.
Mm-hmm. Great. And if you look at the genetics for these activator inhibitor proteins, do you see that the sequence is different for the two different species?
So yes, so there's basically, Josh would be the best person to answer this question, but there is a lot of genomics that's going on in Josh's lab and Ariel Cooley's lab, our collaborator, and others. And so, yes, the idea is that we either know already or will learn in the course of the empirical part of Emily's work, various details about the different alleles that are present for the activator Nigan and the inhibitor red tongue.
Okay, thank you very much. Those were all my questions for now.
Another thing is that, plant cells have a large vacuole, right? So there's that aspect of what's going on microscopically is not sort of in the picture yet with the reaction diffusion equations that we're using. So it's, right now, so I'd like you to just sort of trust me that the more realistic gene regulatory network modeling is something that we're doing in parallel. But right now I'm trying to just explain sort of the logic behind this question of how does a new phenotype emerge from the cross? And that's something that seems like a relatively generic Turing system could give us some insight into. So let me find my presentation again. All right, so are you able to see my screen? Yeah, okay. All right, yes. So basically this introduction is really just meant to give you enough background to motivate why I'm interested in 4x4 matrices that have a block structure, a 2x2 block structure, and I'm interested in their stability properties. I'm interested in their trace and their determinants and how those relate to the stability properties of the individual blocks. So does everyone get that part of it? The main point here is just, wow, I because of Emily's work, I got interested in the determinants and eigenvalues of just general properties, matrix analytic properties of 2x2 block matrices. And because it seems like that's going to be the key to understanding the instabilities in the heterozygous case. Okay, so now I'm going to transition into just some of the things that would be sort of background about determinants and also some things that I've learned and might be new for many of you about determinants. So the first thing to say is that what a determinant is, a scalar value that can be computed from the elements of a square matrix, and that determinant gives you a lot of information about properties of the matrix and properties of the linear transformation that a particular matrix describes. So we will, various times you'll see people call this determinant DETA, or they'll use like absolute value signs around a capital letter for a matrix that will indicate the determinant. And as we all know from high school, right, the determinant of a 2x2 matrix that has elements A, B, C, and D is A times D minus B times C. You have this sort of cross multiplication thing that you do. And for a 3x3 matrix, there's a sort of recursive method of calculating what the result is that is referred to as a Laplace expansion. Now, one of the things that I feel like pedagogically is important to understand is it's sort of just the question of like, why is this number AD minus BC the determinant of this 2x2 matrix? And I think the best answer to that is that really the most important thing that you want to know about a matrix is whether or not it's singular. Okay, so a matrix that's singular does not have an inverse. And a matrix, if you just sort of say, okay, I'm looking for some scalar function of the elements of the matrix that's going to signal to me that my matrix is singular, right? So let's just say we're looking for a function of A, B, C, and D that's going to end up being 0 when the matrix is singular. Well, what does it mean for the matrix to be singular? One of the ways that you can think about that is that this matrix A has a column AC, that's one 2-vector, and there's a second 2-vector BD. And the matrix will be singular if those 2 vectors are pointing in the same direction in 2 space. So that would mean that the vector AC was a scalar multiple of the vector BD, right? Now, so that means that the 2 vectors are linearly dependent, they're pointing in the same direction. That'll cause this matrix to be singular. So if you expand this, you have A equals alpha B and you have C equals alpha D. And then you do a little algebra and you find out that if these 2 vectors here are linearly dependent, then that is going to happen when A times E minus B times C is 0. So the function for 2x2 matrices that we want to use for this determinant, right, this magic operator that's going to work on the elements of this matrix is evidently AD minus BC. What about a 3x3 matrix? Well, one of the ways that you can do this is a good exercise for undergraduates. Start with your general 3x3 matrix and then do Gaussian elimination. So do elementary row operations on this matrix until you clear out the 2 zeros below the A. And then if you think about this matrix can only be singular it's singular precisely when this 2 by 2 piece down here in the bottom is singular, right? These 3 vectors are only going to be linearly dependent if these 2 vectors are, these two vectors are linearly dependent. So what do we do? Let's plug in those values into our expression for the determinant, which ought to be 0. You chug away and you find that the function that ought to be called the determinant for a 3 by 3 matrix is this one here at the bottom. Okay, which happens to agree with this thing that you've all learned about Laplace expansions. Happens to be the correct function. So really, I think this is this is one of the definitions of what, this is a property of a determinant that's really important. And it's probably better to start from this idea in terms of just what is the determinant than it is to just start from these this crazy sort of cross multiplication process that isn't really well motivated. Now, digging into what the Laplace expansion really is a little bit, right? What you're doing is you're picking a row and looking at the elements of your matrix in that row, and then sort of crossing out that given row and column, and then calculating the determinants of the minors associated with that element. And so this is how we get this expression here. It's A, which is the blue entry here, times the determinant of this piece, and so on. So I'm going to be using pictures like this to represent things that we're doing with determinants a lot. So I wanted to illustrate this one where we really do know where it's, everyone knows the answer. So when you do a Laplace expansion of a determinant, right, you can do it for a larger matrix, and you can do it for any row or column. So here I'm expanding the determinant of this 4 by 4 matrix using the second row, right? And then each one of these minors is 3 by 3. So we're going to have 8 terms that comes out of each one of these, and so there'll be a total of 24 terms. And the only thing you have to kind of figure out and remember is what the sign ought to be when you whether you sort of add these to one another or subtract them from one another. And the mnemonic for figuring that out is to just think of it as the matrix is a checkerboard, and the light elements will correspond to positive and the dark elements will correspond to negative. So this term here is negative because the blue element that the first element of this row that we're using in our Laplace expansion is sitting on a dark square in the checkerboard. That makes it negative. Okay, so what we've learned so far, let's do a little sanity check. Who wants to tell me what the determinant of this matrix is? Just do it in your head using what I've told you. And I'm going to count to 10, and if no one gives the answer, then I'm going to just show you all. Can you do it, Leah? Jump in, come on. Okay, so the way you can do it.
77 or negative 77, negative 70.
Negative 77. Okay, I got positive 77, but here's the, that was pretty good. So let's see. So the first thing you do is, if you expand along this row, right, you're gonna get a lot of zeros. So we'll do that. And so we remember that we had a 7, right? 7 times something. Is that supposed to be a positive or a negative 7? It's this one, right? So it's negative 7. Negative 7 times this determinant of the 3 by 3 matrix of what remains. And I can expand that one and use these 2 zeros here so that I get 11 times the determinant of 2 times 1, or the determinant of this matrix, 2, 1, 1, 1. Okay, and that's 2 times 1 minus 1 times 1, which is 1. So the only question is, was this element that I used, 11, was it supposed to come in with a positive or negative sign? And it looks to me like it's coming in with a positive sign, right? So what did we get? Minus 77? or 77?
I think it's negative because you've got the -7, +11, +1. Is that right?
Yeah, could be negative. There might be a sign error here. Okay. All right. So now one of the things that I found in my reading that was what was really interesting to me is, you know, there's these different ways that you learn in school of how to calculate the determinant, but one of them that has been pretty much forgotten that was developed by Lewis Carroll a long time ago is this method that's now called Dodson condensation. Okay, so I think this is going to be new for everyone, but it's really a nifty way to calculate a determinant. So think of your matrix as being these balls that have numbers on them, and I'm just going to use integers for argument's sake here, and stack them on like you're sort of stacking oranges or something in a grocery store on a bunch of 1 balls. Okay, and now what we're going to do is whenever we look at our matrix, we're going to take every 2 by 2 submatrix, and we're going to take that determinant, A times D minus B times C, but we're going to divide by the value of the ball underneath. Okay, so the balls that we started, so we don't use these ones on the outside. They're just sort of holding up the marbles or the oranges. And right now, all of these spaces, these balls that you sort of see peeking through have the value 1. So really, we're just calculating a bunch of 2 by 2 determinants, right? So 2 times 1 minus 3 times 4 is 14, and so on. All right, so I just calculated all those submatrices, all those 2 by 2 determinants and filled them in. And now I'm going to do it again. For example, here, I'm going to just have 14 times -6 minus 5 times -22 divided by 1, and that's the 21 here. Here, when I do this determinant, I'm going to divide by -4 before I put the answer of -105 in. Okay. So you keep on doing that until the very end. In the last step here, I have 21 times 36 times minus the product of -8 times -105 divided by -6, and I get 14, which is the answer for the determinant. So I don't, I just, when I discovered this in my reading, right, the way it went was I was trying to understand how can we express determinants in terms of 2 by 2 subdeterminants. I only wanted 2 by 2 minors in the calculation, and I stumbled upon this really this sort of forgotten result by John Dodson, who's Lewis Carroll. Okay, so let's take a second now and just you know, we've talked about the mechanics of calculating determinants, right? But what about these amazing properties of the determinant? That, you know, what is it actually encoding about this matrix and the linear transformation described by the matrix? So can people just throw out some, what is the determinant actually telling you about the matrix? So we said first, we already got the fact that when the determinant's 0, that means the matrix is singular. It doesn't have an inverse. When the determinant's 0, it means the columns are linearly dependent. Right? How else does determinants come up? Any thoughts?
When finding eigenvalues and eigenvectors, did you say that?
No, we haven't really talked about that yet. But so yeah, determinants, they're really coming up in mathematical biology a lot of the times because when you're thinking about linearized systems, right? The determinant of the matrix is one aspect of the matrix that is going to contribute to whether or not what the eigenvalues actually are, right? So the determinant is the product of the eigenvalues, and in simple cases, when you have, for example, just a 2x2 matrix, right, then your stability condition for this sort of linear system is just that the trace has to be negative and the determinant has to be positive. If you think about it, if you have two eigenvalues and their sum is the trace and their product is the determinant, then in order for both of those eigenvalues to have negative real part, these properties have to hold for the matrix. Okay. Okay, I'm gonna skip through some of this background material. Keep things moving. Another one is, you know, so this point that we're making here is that, you know, it's the trace and the determinant of a 2x2 matrix that are influencing the dynamics of the linear system. So that's one of the reasons why we're into determinants. People sometimes learn about the area of this parallelogram that you get when you think about your transformed unit vectors. So these are your unit vectors, you multiply by the matrix and you get these other vectors, and then that area turns out to be the determinant. It's a signed area, but it's the determinant. And then, you know, this is the one, this is the property of the determinant that you know, Emily is interested in and that I'm interested in. And this has occurred in a lot of Zhongping's work as well as Leah's. Okay, so it's the determinant, right, of the Jacobian matrix of the that you can calculate from the nonlinear reaction terms in a Turing type model. That if you want it to be unstable, that determinant needs to go negative, right? So what we're looking for when we do these Turing analysis type, when you do Turing analysis of Turing systems, is you want the spatially homogeneous steady state to be stable without diffusion. So this matrix needs to be stable. So its determinant needs to be positive and its trace needs to be negative. But then when you consider the linearized system and the effect of the linearized PDEs and the effect of diffusion, the matrix that you get there needs to be needs to have a positive determinant. So this is we're going to blaze right through this. If anyone's interested, there's lots of good reviews of this technique, but you linearize the PDEs, you assume ensembles for the solution that has an oscillating spatial and exponential growth in the temporal part. And then when it all comes down to having to have this determinant here be 0, if you're this is actually if this is satisfied, you have an eigenvalue of the system, and so you're looking for a situation where when there's no diffusion, you have stability, but when you have diffusion, you have instability for a particular wave number, and that's the whole story. You basically find out that you can get these types of Turing instabilities to happen if the inhibitor is diffusing faster, sufficiently faster than the activator. All right, but one of the things that we're still a little, you know, vague on is what really is the best definition for what a determinant is, right? So this is a Blackboard from a YouTube video that I watched that I enjoyed a lot. It's writing a whole bunch of properties of the determinant, right? The determinant of a transpose of a matrix is the same as the determinant of that matrix. If you can get upper triangular matrix, its determinant is just the product of its diagonal entries. Here's that parallelogram thing that we were talking about. And then over on the left, you see all these things like, you know, what happens if you transpose 2 columns in your matrix, that ends up changing the size, or what happens if you scale a column or a row, and so on. And one of the things that's interesting is when you read more mathematical, when you read papers written by algebraists about determinants, right? The definition that you get is the, you know, the first thing that gets written down here. This is a Zen determinant movie. Okay, so the first thing that's written down, right, is this. The determinant of the matrix is involving sum over permutations of products of the elements of the matrix. And this is something that might be less familiar to many of you that I wanted to go over. So, for example, this is another way of writing it. So, what we're doing is, what we're going to do is think about a permutation of the numbers 1, 2, and 3. There's 6 of them, and for every permutation, there's an associated permutation matrix. So for the permutation 2, 1, 3, I'm going to put a 1 in the second column of the first row. I'll put it in the second row, I'm going to put the 1 in the first column. In the third row, I'm going to put the 1 in the third column. I think this correspondence is pretty clear. And so if you basically take every permutation of the numbers 1, 2, and 3, and then as you go through the rows of your matrix, grab the appropriate column, the element that's in the appropriate column, and then you pick a sign that depends on whether that permutation is even or odd, then you end up getting the correct set of terms. So this is actually the definition that most mathematicians start off with. And what's nice about this is it makes a number of things clear. So for example, you know, how many terms is this determinant going to have? Well, it's going to be 4 factorial, right? Because that's the number of permutations of 4 things. So 4 factorial is 24, okay? And then this issue of whether the sign should be positive or negative, for small examples, there's a geometric object called the permutahedron that you can walk along, but depending on the number of steps that you take, since each one of these steps is actually the transposition of 2 of your elements, the signs are sort of alternating as you go through here. So 1, 2, 3, 4 has a positive sign. 1, 3, 2, 4 would have a negative sign. But 2, 3, 1, 4, that permutation is even. So this term is going to arise in the determinant with a positive sign. Okay, so this is really, I think, you know, the best way from my reading. That I think this is the way I'd actually want to get undergraduates to the point of understanding this aspect of what determinants actually are. You're essentially taking, you can just think of all the different permutation matrices, the red ones being even permutations and the black ones being odd permutations, and then just take your matrix and overlay these matrices of 1s of 0s and take the element by element product, right? And out comes this homogeneous polynomial that has the right number of terms and the right degree. Okay, so back to our problem. So I was interested in, right, that I wanted to understand about eigenvalues and determinants of block 4x4 matrices. And in my reading of this book, A Treatise on the Theory of Determinants, which is a very famous classical monograph by Thomas Muir, I found this Laplace expansion that I'd never seen before. So I don't know how many of you have seen this, but basically this is saying that the determinant of this 4x4 matrix is the determinant of this 2x2 submatrix times the determinant of this 2x2 submatrix, and so on. So when you look at this one, all of the blue and red here is respecting the horizontal line right? It's respecting the block structure of the matrices that Emily and I are working with. And so I was glad to find this. And I think that understanding this expansion of the determinant might be helpful in our analysis. And this one, this expansion or this rule for determinants was also something that
Hello. I'm pulled 6 ways. I have 2 different
Hey, Eric, you're on.
I turned him off.
Okay, thanks. So this one was cool. This one goes by the name of Dodson, right? This is also developed by Lewis Carroll. So this determinant, right, times the central element is equal to the determinant of these subdeterminants, right? So it turns out that this is called Dodson's rule because this is basically one of the identities that's actually used in the Dodson condensation method of calculating determinants. And the general form for the rule can be written like this. It's a little hard to parse these sorts of equations, but basically, let's look at the bottom row, right? This is the identity. You've got a determinant of determinants here, and then you've got a product of determinants on the left. So you've got the whole thing, scrape off the edges, and then you sort of take this scraping off alternately the first or the last row and column and calculate those determinants. And you get your, that's what this formula, when it's unpacked, actually means. And when you see proofs or expositions of why Dodson's condensation works, it's this formula that is at the basis of it. Because, right, in the 4x4 case, for example, here's, remember this product is equal to this determinant of determinants. So this product of determinants is equal to this determinant of determinants. And so you can kind of recursively now take this 3x3 determinant and also use Dodson's rule on it. And you'll end up with this 4x4 matrix, right, being pretty much just a scaled version of a whole bunch of 2x2 determinants. So I thought this was, the connection to Lewis Carroll was cool, and running into this method of Dodson's condensation as I was pursuing just trying to understand determinants of block matrices, I just it was really exciting to me. This is another picture of Dodson's condensation, right? Here's the ones below it. Here's a 3x3 matrix. And then here's the rule, right? The value put here is this determinant AD minus BC divided by the element that's sort of underneath. Okay, so I'm gonna make a transition, talk about something related, but at a slightly different level. And so it's a good time if anyone wants to throw in a question.
This question's actually for later. Would you mind sharing that video that you said you really liked in terms of different ways of looking at the determinant? It was by Grinfeld, but he has a lot of videos, so I couldn't find that specific one.
Yes, I can share links to those videos as well as, well, I've got recommendations for further reading. There's a couple of PDFs that I've already put on the webpage, and if I guess I can put a Dropbox link or something to a bundle of PDFs as well if you're interested in further reading on the topic.
Thanks.
Sure.
I have a question. So you didn't say the detail about your model, but but it seems like you have one subsystem has a red as the equilibrium, the other has a yellow. So for the 4-dimensional system, the equilibrium is a half red, half yellow, or they are both red or both yellow?
Yeah, so the cupreus and the variegatus strains here are not as patterned as in F1, but they sort of have different anthocyanin levels. Is that what you're referring to?
Yeah, I mean, it seems like if you have a four-dimensional system, so if both UV and U hat, V hat equal to the red, that's like equilibrium.
Right.
If they both equal to yellow, it's also equilibrium. But if you make UV equal to red, U hat, V hat equal to yellow, I don't know, that may not even be an equilibrium. So because eventually you are going to do linearization, so which equilibrium you are going to linearize it. So
yeah, okay, so you're exactly right. So what we haven't really gotten to that part of things yet. So when you in the full numerical analysis of this sort of thing, which is something that Emily's doing, right, is she's going to need to find parameters that correspond to these, parameter sets that correspond to these two different Turing stable solutions, right? Whether it's stable to spatial perturbations. And then when those two parameter sets are combined in a way that is corresponding to the heterozygous F1 generation, then at that point, the idea is that the solution should have a pattern. I mean, that's what's happening biologically. And the question is, can we understand when and how that happens mathematically? But it's not going to be you're not linearizing around, you might not have the same steady state at all. You're absolutely right. Okay. If you have really strong conditions for the symmetry of what's going on, we've actually found the results where you can have your two homozygous parents being identical, actually, in terms of most of the parameters. I mean, but when you combine them, they're still, so how should I say this? Identical in terms of the values of parameters, but formally, the gene products are still different gene products, right? And then when you combine them, that means that the association of one gene product to the regulatory region of the other genome can have a different parameter. The trans interaction can be different than the cis interaction. So there's sort of a simplest case where you have everything is completely symmetric, except that in the F1 generation, the heterozygous case, the trans interactions of your transcription factors are less efficacious than the cis interactions. And one sort of question is, can we analytically or understand which of those trans interactions being suppressed in the F1 generation is actually most likely to lead to the pattern in the F1 generation? And I think that that's likely, so Emily's already got numerical examples of that, and I think we'll be able to get some analytical results related to that simple case because of all the symmetries. But for example, you might find that it's enough for the trans interactions of the inhibitor to be suppressed. And that's enough to have the variability in the F1 occur. And then the other thing that's happening is there's the F2 generation where experimentally you see a wide distribution of responses. And that can be looked at in the model too, at least numerically, because there's sort of the logic behind how the parameters should be shuffled to correspond to recombination. So that's where we're hoping to go with that. Now, let me just, to finish the presentation, I want to talk about one sort of interesting aspect of this that came up, which is that if you sort of start formally with just indeterminants for the values of the matrices that you're trying to calculate the determinant of, and then you run through the Dogson's condensation, you end up getting these expressions that my pictures were alluding to, right? Where you have these products of determinants, but then you're dividing by certain elements from the previous row. And when combinatorial discrete mathematicians, experts in combinatorics, were looking at this, they realized that, like, in this simple case where it's the determinant of a 3x3 matrix, you actually see that you end up with you end up with more terms here than you would get if you were just looking at permutations. And if you tried to figure out the terms that involved the division here, those terms would correspond to situations where you had you sort of had something like a permutation matrix, but you had negative entries in certain places, right? So there's basically, so I guess what I'm saying is that the understanding of mathematicians about why Dogson's condensation actually works had led to research on something called alternating sign matrices. So an alternating sign matrix is a matrix where every row and every column starts with a 1, but you can have -1s as well, as long as the -1s are alternating. So as you go left to right here, I got 0, 1, -1, and then there's the next one. So you can have the zeros and the ones anywhere, but the -1s have to be appearing kind of internally, and they have to be alternating with the positive ones. So it turns out that this particular type of matrix, an alternating sign matrix, is sort of the key to understanding the combinatorics of what was going on in Dogson's condensation. And so people about 20 years ago started digging into this, and you know, you can, for example, just in a computer, see that for the alternating sign matrices of sign 3 are just the normal permutation matrices plus this 1 in the center here that has a -1. And it turns out that in the polynomials that arise here, there's going to be 2 terms that correspond to that alternating sign matrix, and they cancel. And that's why this expression ends up being the same as what the determinant that you would get just by using the permutation matrices. So that's what he's writing over here on the board. Each alternating sign matrix appears in this polynomial expression 2 to the power of the number of -1s times. And since it's occurring an even number of times with alternate signs, those terms all cancel. Turns out that there's 42 alternating sign matrices of size 4. And here was, it's an interesting story that people basically discovered this idea of the alternating sign matrix. They, through some brilliant guesswork, arrived at this formula for the number of alternating sign matrices you would have of any given size. And then proving this combinatorially is a really interesting story in sort of the history of mathematics, because there was a connection to something called plane partitions, these sort of blocks that are stacked. You have a certain number of blocks and you sort of stack them in the corner of a room. How many ways can you do this sort of thing? Alternating sign matrices. And then there was also a connection to something called square ice, which is a, if you're familiar, like, with the Ising model, a 2-dimensional model of magnetism, this is a 2-dimensional statistical mechanical model of ice. And this alternating sign matrix, every alternating sign matrix will correspond to one of the configurations of this in this statistical mechanical model of ice. And the mathematicians eventually found the physicists who understood this model, which turned out to be one of the small number of exactly solved models in statistical mechanics. And that connection allowed the proof of some of the conjectures associated with these alternating sign matrices. Okay, so the bottom line is, takeaway is, first of all, this method of calculating determinants. Someday, hopefully next time I talk about this work, I'll be showing you all the analytical results that I obtained using my new understanding of determinants. And the further reading that I'd recommend, if you're interested in determinants in general, this book was actually pretty accessible. And this one, Proofs and Confirmations, is the story of the combinatorics of alternating sign matrices. And, you know, Lewis Carroll sort of is in the picture as this story starts off, but then it moves into, it's a really great sort of history of the mathematics of this particular combinatorics problem. And then if you want a more mathematical understanding of why this Dogson's condensation method works, I think that this paper is the most accessible one that I found so far. So thank you very much for listening.
Thank you.
I see lots of claps. They're not very loud, but they're visible. Any questions? I feel like one of the things that's interesting to me about this is that, I mean, the Turing patterns and this way of thinking about developmental biology is, you know, there's definitely a lot of room for criticism in terms of how idealized it is. On the other hand, if you find a good review that's been written recently about Turing, analysis of Turing systems, you know, there are examples where this sort of thing really does seem to be happening. There's other examples, there's other ways that patterns can arise, morphogen gradients, you know, and a whole host of other things. So when you have stochastic sulfate decisions, there's all sorts of different ways that these things can arise. But you know, if it is known in like trichome, little hairs that grow on leaves, that there's lateral inhibition that's occurring in that system. And so it's not out of the picture that it might actually be Turing instability that's leading to the heterogeneity in the patterns that people see in the Mimulus system. And the idea of like not asking the question, can I get a Turing instability by having a reaction diffusion system and then sort of adding diffusion? I mean, having a reaction system and then adding diffusion, but instead ask the question, can I have two systems that are Turing stable, but when they're genetically interacting, when they're in the same genome, right? Can those cross interactions actually lead to the instability? I think that's a pretty interesting question, and I'd be interested, jumping, if you've ever heard of such an analysis before, but as far as I know, asking the question in that way is pretty new.
I have another heard about that, but usually your system seems like you have a bistable structure because you have two subsystems has a stable equilibrium. Yeah, anyway, for the two-dimensional system, if you have a bistable structure, that's more likely to have a Turing pattern. Now, of course,
Yeah.
a Turing pattern can occur with just one stable equilibrium, but when you have a two, usually it's more likely. Yeah.
So from the perspective of the biology, there's actually no evidence that there's actually bistability, right? Because the two homogeneous steady states, spatially homogeneous steady states, are systems that have different parameters. They're two different variants, right? And so the issue is though when they're combined, right? What happens? But you're right. I've you know how when you're doing research, you always try to take the simplest thing that you actually can understand and work that through first. Well, if you just have a self-activating, like an activator that can activate itself, and that's all you've got going on, you can have situations where the homozygous case has monostability, and the heterozygous case has bistability. I thought that was kind of interesting.
Greg, could I jump in and ask a biology type question?
Oh, no.
Because I can't get away from so much of this, like what you're going to apply mathematically should be constrained by what is kind of happening biologically, and that will rule out or rule in certain things. And so much of this could be known if really it's that simple that transcription factors are just moving around through these like really large gap junctions. Has anybody just done a simple immunocytochemistry, tag a protein, the GFP? You could watch this happen.
Yeah.
So
In a fairly simple experiment. So either take snapshots in time or look at it with a GFP fusion protein tag, and that will rule in or rule out so much of
Yeah,
math.
and there's also genome science methods of actually trying to detect the interaction of the transcription factors with the regulatory regions, right? Like the ChIP-seq and stuff like that.
Oh yeah, that's, yeah.
So that the
So we're working on doing that. We're breeding the flowers so that we can do some of that ChIP-seq data. But part of the problem in terms of tracking the Turing patterns from an experimental standpoint is that we don't actually know during what developmental stage this is happening. So part of what we're going to be doing is looking at buds versus fully developed petals, because there seems to be some evidence that the patterns are showing up on the petals early and then they spread, or the alternative hypothesis is that the patterns sort of fill in as the petal is growing. So when we have this Turing pattern on a physical space that's changing, the 2D plane is actually growing as the pattern is developing. We're not really sure how to
That's development.
yeah, well, so the experimental stuff is I'm working on learning it. I don't know it very well yet, but that's what we're working on.
So the easier thing to do would be to do a few, before you do a fusion protein live imaging, just a few snapshots in time at a few relevant stages to get an idea.
Yeah, we have a few snapshots, but trying to determine how much of it is genetic and how much of it is, you know, the subtle variation between two different flowers on the same plant. There's a lot of work that needs to be done before we can really get a handle on when this is happening and where it's happening and how the differences happen between flowers.
My impression from those preliminary results when we looked at the developmental stages, and so you're pulling off the petals at different from one plant at different times and then looking at them, it's pretty amazing how how similar they are. I mean,
Really?
Yeah,
Oh.
yeah. I was
Wow.
very concerned about this, but you look at it, and if you had two different plants in a range of developmental stages, and you had to sort them, you'd get it right every time. So
That makes it much easier for you. Not that it's easy, but I mean, when I look at the amount of individual variation in some systems, it's just an epigenetic mess.
And then in terms
actually,
of ruling things out by doing, by initial empirical work, Ariel Cooley has done this, and a collaborator of hers has done this sort of thing, where from the perspective of RNA silencing. So is that right, Emily? Did they knock down Negan, or did they knock down red tongue? Do you remember?
I don't remember offhand. I know that there's an experiment where they made a red tongue knockdown mutation, but that was the one, Yuan was the last name.
Yeah.
And I don't think Ariel was involved in that, but she has worked with them in the past.
Is it? I'm not sure whether it's. It might not have been RNA, antisense RNA that was used, But there was a, there's empirical results where you can knock down the inhibitor and all of a sudden you see more spots, or it's brighter at least, and things like that. So there are at least preliminary results along those lines.
So just quickly, how different are the amino acid sequences between the two species and the two, like what are we talking about? How different
The homology.
are those proteins?
I don't
Yeah,
know.
at amino acid level.
Yeah, I actually don't know if it's known.
Okay, I just wondered.
Yeah. And so the
Seems like something that should be known, but I don't know that we've done that experiment yet.
Well, you don't, yeah, I mean, you should be able, because the genome has been sequenced, right? So it's a simple blast search that you could get this from.
yeah, that you're comparing
just
two different
curious.
varieties, right? So it might be some subtleties based on which Mimulus genome was the one that was sequenced.
You should be able to get some idea.
Yeah. I think that would be
Yeah, I mean, that should be pretty easy to figure out. Right, thank you.
okay. Thanks for everyone's attention and questions, and see you next week. Bye-bye.
Bye-bye.